missed some things on the first week, but can catch up. And Madison was here last time. So we have a total of 6 students, and that's great, absolutely great, because you can see it's a capstone, and a lot of this is going to be trying to be us helping each other, especially me helping you on your own project like that, okay? And so, I am recording these, all these meetings, and if you miss class, we have a requirement that you have to take notes and send me pictures of your notes, just so everybody keeps up, okay? And so what are we doing this week? And so you're just always working on your capstone journal effort. And then we on Monday talked about lifespan. And something I haven't mentioned is that we want to follow up on that. So we talked about it, but we want to keep that in mind as one of the topics that we learned some about. And you could ask questions about it this Wednesday, today, and say, okay, I still didn't get that thing we talked about on Monday. And also, you can bring it up throughout the course and say, how does this compare to that Monday stuff, like that, okay? So lifespan is something we want to have learned something about and have it on an ongoing, in our mind, like that. So we talked about why would that come up in our course, why would it come up first, like that, right? Today I'm going to talk about chapter 1 from the online textbook reading from the Gilbert textbook, and so it's on so everyone's on electronic reserve. And on Monday we'll sit in a circle and we'll all talk about chapter 1. And so as you learn about chapter 1, you should be writing down ideas, questions, and additions that you can make to our Monday discussion on this topic. And the topic is the introduction of a variety of examples of this phenotypic plasticity. So the first half of our course is about normal differences, and we talked some about abnormal differences. That'll be the second half. And we did that primarily just to show the flow of our course, but also try to help you start to think about possible capstone projects. So again, today, and that's going to be our plan. We do this kind of hopscotch or leapfrog thing where on Wednesdays I'll essentially a lecture on the next topic, and then the following Monday everybody's going to contribute in a roundtable discussion about it. And you can bring anything, right? Any resources, and they could be as parts of your capstone project you're working on. They could be from another course you've had, or another textbook, or a website, or video. And if you have pictures that you want to share, that you find, send me those so I can pull them up on my PowerPoint. And we just want to be involved in it like that. Okay. All right. And so that's what we'll talk about today. And so again, number 4 here, prior to next class on Monday, send pics of 5 things that you could add to next Monday's discussion. And all along the way, write down stuff that you're confused about or need help with, like that. Okay, so that's where we're at. And that's going to be the ongoing flow of our course. And we've really got 2 goals in mind all the time. And one is you're working on your capstone project, and you don't know what it is probably yet, so you're writing in your capstone journal, which is one of the 2 shared documents that I gave you at the start of the semester, and you're adding to that, and you have to have stuff in there, and you never delete stuff. Like if you say, I changed my topic, so I deleted everything. That doesn't get deleted from your capstone journal. And because that's part of the effort, is to gravitate toward a topic, and it might take you a month, but we're working toward that topic. It might take you a day. You might have walked in with intention of studying, something like that. And then the other thing is what we're doing in class. And so what we're doing in class is going to be assessed by 2 exams, a midterm and final. Those are going to be open-ended questions about these topics. So the question could be about lifespan, and we talked about that some on Monday, and how that could be a question, and what would be like not a question, because it could be both a broad question about what you learned about lifespan, but also a specific aspect of it, like that, like maybe specific aspect about the nematode. And so there's some coaching about how to learn in the class meeting to prepare for the exams, okay, like that. So does anybody have any questions, comments, confusion about anything at all? And you can bring those up at any time don't worry if they seem to be out of place. It's my job, to help, put it in context for the class. Yeah. All right. So here's just something that I look back over the years that I've taught this, and so here's just some wild things about how I thought students might pick topics. And so these aren't all the topics. I have a couple different slides of it, right? But these are things that came up that students pursued in their capstone projects. And I didn't write down the specific topic they were pursuing, but I made it broader. So believe it or not, that's a topic for our course. And a student did a great job. And the student was doing an internship for Portland Water District, and was out at the Casco, or at the Sebago Lake water source of drinking water, and learning about that, and learning about potential contaminants in the water and how those could affect developmental health in the people who drink it. And then also went to the wastewater plant and looked at how that could impact aquatic life and stuff that's going out in there. Here's an example of that. Now they can detect lots of different pharmaceuticals in wastewater, and those just go out in the ocean, and are they at enough a concentration to affect things like that? They're also starting to detect that in Sebago Lake. Not just like kind of traditional pollutant type contaminants, but some really specific ones that people are concerned about, like that. And so, it was a great project because it was kind of out in the community. There were experts in the area, like that, and the student went on to work, as far as I know, to this day, for Portland Water District, because they were just like, what are you doing? What's that class about? And they were fascinated by the student and the path she was on. Then any aspect of agriculture, and there are a lot of different things in agriculture, and in the context of a lot of either organic farming or this kind of modern factory farming type work. Then a student was going to be a school teacher, and so used the topics in our course and was pursuing sort of like kind of like public health and public education about some of these topics like that. We mentioned BPA and phthalates, and so that student worked on these phthalates, and it was very interesting because one of the things they were pursuing that they wrote about in their capstone project was that there's a real issue about socioeconomic status. So lower income folks tend to be the last people to hear about health concerns, like say, for example, lead paint, like that, and be aware that you want to keep your little kids away from chips of lead paint in their old building like that. So, just really interesting, and matched up their interest, going to be a teacher like that, and fitting it in, right? And these are interesting. I picked these two and put them here because they seem so different than what we're up to, right? We're up to looking at like a little nematode worm and understanding, the molecular genetics of how it extends its lifespan like that. So these are totally legit. Then a student worked there used to be a toxicology research lab here, and she was doing, lab work in there, so she wrote about her topic there. And then a few students have worked on specifically being interested in wildlife and wildlife populations. One student in particular connected with this company in town called Biodiversity Research Institute. They study wild bird populations, and they're monitoring them, and we actually have several former USM students, USM graduates, who work there. And so they're very, very welcoming. And that is just here in town, and they actually, monitoring different things like lead and mercury in wild birds and other molecules. You're doing that?
I'm doing that internship at BRI.
Oh, yeah, that's fabulous. And so there's a worker in the toxicology lab. Are you in the toxicology lab?
Mm-hmm.
Yeah, so Cassie. Does Cassie still work in there?
Cassie's one of my supervisors.
Yeah, so
she's great.
Yeah, Cassie's fantastic. So so Madison knows about that, right? And that could be your topic. right? And bringing, not having it be, oh, that's what I do at work, but actually in every case, pushing hard to bring it into the context of the topics in our course, like that. And so students have studied the health of wild populations, right? We know about BPA in human health, but those endocrine disruptors are out big time in nature, and there's a lot of those kind of disruptions. So probably kind of a second half of the course for the topic like that. Yep. That's fascinating and fantastic medicine. And so then here are just some topics that I have listed because they're really part of our course that will come up. So, and again, I'm recording this, so, if you want to go back to these, you don't have to scramble to write them down. A lot of students are interested in epigenetics, and we'll talk about that. This semester hasn't come up yet, but that's this process of DNA methylation. So that's this a common molecular mechanism to modulate the genotype to have phenotypic plasticity. And so we'll have examples of that, and there are just some students that I'm fascinated by epigenetics, and there's a lot of epigenetics in our course. And so, just some topics that will kind of flow through in our course. And then here's more of topics another time that I put on a syllabus that students had done. So different topics, and I won't go through each of these because again, you can look back at them. But again, it's just interesting. One of the things we want to do is make sure that you might start out with a pretty broad topic, but eventually, kind of narrow in on it so you make it a realistic, project, right? Sometimes students have caught something that's kind of too big to really turn into I'm going to get it done this semester. Yeah, like that, okay? So, whatever your interests are, we're trying to match it up. We're not trying to, force you to take a topic that's really different than the stuff you're interested in, like that, a student who is going to be a teacher, we fit that in, okay? So here's just a couple of follow-up slides. Just want to follow up on this, and this, is a blizzard of stuff, but I pointed out last time that this is a cell in our body, and most multicellular animals. And here's the cell membrane, and here's intracellular signal transduction pathways. There's the insulin signaling system. And so we're going to have inputs from the outside, and they're especially modulating TOR and FOXO. Those are just key words that come up. And we talked about them, and one thing we think about when we think about FOXO is we think about it's a big changes in gene expression. And so when FOXO gets to go into the nucleus and do its function, it's going to extend the lifespan. A memorable way to think about that is this. We have all the same genes, but we have different alleles, different hair color and stuff like that. There are different alleles of FOXO in humans. And if you go to these families in some communities, there are a few villages here and there in Turkey and China and stuff, and they'll have a set of people that just live remarkably long. Like a lot of people living over 100. Those people tend to correlate with specific alleles of FOXO. Different hair color, just like that, right? It's a heritable trait, and that would mean that there would be some variety, some diversity in the population. And so, FOXO is a really critical thing. I just wanted to talk a little bit about, so that's what we think about FOXO. Here's what a good way to think about this TOR. So I talked on Monday that TOR is really, when it gets to do its job, it's going to increase ribosome number and activity because it's going to try to make things grow. And so you can find ortholog of FOXO in every animal. You can find orthologs of TOR in every eukaryotic cell, including single-celled eukaryotes. So we'll just talk about that for a moment. So the yeast that causes bread to rise has an ortholog of TOR. And so in a single-celled eukaryote, this is what you have going on in a single-celled eukaryote. So it's kind of a composite figure from a review. But the individual cells in the yeast that causes bread to rot in a single cell eukaryote, those individual cells are making direct responses to availability of nutrients. So that's showing here that there are the nutrients and that this cell is going to decide on its own if I've got the stuff I need to grow and divide. And a big target of that pathway of growth is TOR. So it's great to think about that, right? Our single-celled eukaryotic ancestors could grow and divide or not. And their big thing would be, I gotta see some good quality nutrients, and if I do, I'm gonna grow and divide. And to grow and divide, what I'm gonna do is I'm gonna be just cranking up the ribosome activity. There's more to TOR than that, but it's a big part of what it means to grow. And where's that coming from? From our single-celled eukaryotic ancestors. So all plants and animals on our planet have this TOR mechanism, right? Then, think about this, right? So then multicellularity evolves. So we got multicellular animals, and you and I, we no longer, our individual cells no longer make direct responses to availability of nutrients. You know, in general, we typically have plenty of nutrients going around in our blood. And to be a multicellular organism, what's happened is there are growth regulators, hormones, and signaling factors that are going to be modulating the body's plan. And so that's where we get a complicated figure like the figure that I just showed, because we really have two stories going on, kind of an ancestral one where individual cells are making decisions, and those, although the cells don't make those decisions anymore, they still have these components within the cell. They're playing that role. And that role, that ancestral role, has been integrated with this more like hormonal style signaling, coordinated multicellular signaling like that. So, you know, again, we want to dive into this and hang on to this because we want to have it a framework for learning about lifespan and also other topics come up. Okay? So just following up a little bit on that, you know, from lifespan, right? And we want to keep doing that as we go forward. So chapter 1 is really an introductory chapter, and it's at the level of the organism. So when we're talking about Monday, we'll be like, oh man, this is a crazy thing that happens, right? So the book picks some specific organisms to introduce the principles. And the book is explaining the principles, but it could do more. It could do more to say, why did they pick that example? Because they're picking, very intentionally, the author's picking very specific examples. So I picked a specific example, that little C. elegans worm, because it extends its lifespan. So we can pick on one species that has this diversity of life that opens the door for things like FOXO, you know? And well, what's this FOXO? Human gene FOXO is probably not very important, you know, like, well, it is, right? So that's chapter 1. Chapter 2, the author dives in, and that's what the basis for ecological developmental biology is we know so much about the developmental mechanisms at the molecular and cellular level within an organism. Like I mentioned, hormones. Oh, we know a lot about hormones. So we'll have an example, and in chapter 2 we'll say, you know, what's going on with that example is we know a ton about what would be going on in it. So even if it's a really unusual critter or plant out in the environment, you know, a developmental biologist would see this phenotypic plasticity and would be like, yeah, I bet I know what's going on there. And I mentioned that those people did that with, there's a historical group of birds on the Galapagos Islands called Darwin's finches, and a big pivotal thing in Darwin's thinking and evidence for evolution, where we have slightly different species on the different islands of the Galapagos, and some specialize with a husky beak on chomping up seeds, and others with a thin, narrow beak put their beak into trees to get out little insects like that, you know. And I mentioned that some developmental biologists are like, we'll just study the embryos inside these various finches' eggs, shells, you know, like that, chip them open, and we'll look at gene expression in them. And they quickly found proteins that could modulate that. the shape of the beak. You know, like, how did they get mutated to have these different beaks? And they're like, well, we have very good hypotheses of how that could work. Another example that's very fascinating is, I haven't really read these papers, but several years ago, some people did the same thing with a duck and a chicken, and they could just figure out how to convert a chicken beak into a duck bill by just modulating gene expression in the embryos, of course, in that beak bill thing, and change them, change them around like that, right? And so, fascinating in chapter 2 when we dive into the mechanisms like that, and that's where the disparate, the really seemingly disconnected examples in the first chapter, they all come together like that. And then the third chapter is a story that really drills into a kind of a set of examples. And the third chapter is about how the microbiome is part of the environment, and the microbiome modulates development in lots and lots of examples. So it's worth a chapter because it's just such a hot new topic like that. And it turns out that the examples that these folks who were studying phenotypic plasticity in these various odd organisms, those were the pioneers in understanding how the microbiome is significant. And you have a microbiome in your gut, and maybe you take some probiotics or whatever, all of that started by these people studying topics from our course. And so we'll learn about that chapter 3. So that's what we're rolling forward. So chapter 1, it's kind of a it's kind of a nature walk. So enjoy that, look at these examples, learn the terms, like that, diving in like that. So here's a super huge term in normal plasticity, is that normal plasticity is going to either be a polyphenism or a reaction norm. We just have to have really good working conception of that. And a polyphenism is going to produce discrete forms. The reaction norm produces a continuous range. And we'll tend to talk about polyphenisms because they're good introductory examples where you can see, I can see how those are different, and I can see how you would have to have some kind of hormonal switch. Reaction norms are kind of unlimited. They're just kind of unlimited. There's just so many that you don't even think that's an example. But there are many, many, many of these, either polyphenisms or reaction norms. And so what we'll do is we'll pick one. So here's the book, and the book is introducing these in chapter 1. And we'll pick one, and we'll say this one. And we'll just describe, and you can find examples like this in chapter 1 or online like that. Bring them to class and say, here's something that happens pretty wacky. And we'll say, okay, we've got that example. We'll hang on to that and move into chapter 2 and 3 and other parts of our course like that. So here's a tropical fish, and various species of it live around Earth, and they are all coral, and it's called a wrasse. And this particular species is showing its reproductive behavior. The colorful fish is a male, and the male kind of herds around a group of females, and the male really aggressively chases other males away. And I don't know how long ago they came up with the term, but they refer to the group of females as a harem, like that. A group of females like that. Okay, so that's a setup. That's how these guys, court and reproduce. Is the male is just trying to herd around a group of females, and around the coral reef, and other males are trying to chase them away and stuff like that. Okay, so here's the phenotypic plasticity. Okay, so if we remove that male, the largest female turns into a male. So that's the phenotypic plasticity. And it turns out that many, many fish species change sexes naturally in a whole bunch of different ways, but this is just a good one to start with. And so the largest female. So this is about the phenotypic plasticity of the largest female. Now, it's amazing because these are vertebrates and we're vertebrates, and all vertebrates undergo sex determination and differentiation the same way. It's hormonal, and in all vertebrates, females are produced by having the estrogen hormone and males by testosterone. So we're saying that that largest female has been making estrogen, producing eggs like that, and it's going to change and start to become a male. Okay, so what does it mean to be a male? So one thing is that the behavior is going to change, right? The largest female has different behavior than the male. The largest female, if another male is in the environment, doesn't have to fight and chase that male, but the largest female, in the absence of a male, will start to fight and chase these males within a day. So behavioral changes within a day. And then color changes is another thing, right? So different pigments, and that happens within a week, like that. Then, kind of the surprising thing for us, because we're mammals, is they start to produce sperm. And that's surprising to us because we have internal fertilization. So we have very different sex organs between male and females in humans and mammals. Fish, like this, have external fertilization. They just release the eggs or sperm into the environment. So no womb, no penis, just an opening to release them. So all the fish has to do to change sexes is just changing whether they're producing sperm or egg cells, like that. And they're both haploid cells. They're not that different. It's just a question of how does the germline stem cell differentiate to make an egg or a sperm, like that. And so, it's a dramatic change like that. And it also, we'll talk about this, it also brings up immediately, in our healthcare today, it's possible for a human to have injections of either testosterone or estrogen and change their secondary sex characteristics, right? And that's, I know, a big social and political issue, and we're not addressing that. We're addressing the medical reality of that, And so it's fascinating because it has relevance for human healthcare today. Yeah.
Do we also see in groups of fish or other creatures a change from male to female, or is it typically just female to male?
I'm pretty sure in fish it goes either way. Do you know that, Luke? I think. Yeah.
Off the top of my head, clownfish go male to female, and they'll be the larger one will be the female. Similarly, it's when in the absence of
In the absence of a female change.
present. Yeah.
Understood.
Yeah, I'm going to say that there are some fish species where when they're juvenile, the entire population is one sex, and it's only when they reach sexual maturation that we see two sexes.
Is one more common? Like one change from female to male or male to female, is one of those more common than the other?
I don't know.
Okay.
Yeah, it's just in.
Maybe I'm getting too nitty gritty with it.
Well, no. We want to get nitty gritty, and so yeah, but just to finish up that thought, I'm pretty sure that when there are fish that develop as one sex, I'm pretty sure that it could be either sex. So a whole species is a juvenile all male, or a whole species juvenile all female, and then they're going to mature and produce both. And so that's the phenomenon like that, and it's a good example of developmental plasticity, changing sex like that, right? There are lots of things that change. One thing that we'll talk about is the idea of the changes in the behavior, and are there developmental mechanisms, what's happening in the brain to do those changes like that, okay? So then also you'll see chapter 2, so not really for this week, not coming Monday, but for chapter 2 we'll say, okay, well, what is happening inside these animals? What is the story of vertebrate sex differentiation? So I already mentioned testosterone and estrogen. And so just to add a couple slides like that so you see, where we're going with this, because this is where it will pull a lot of things together, is just to show you this, that there's testosterone and there's estradiol. They're very, very similar molecules. You can see the difference, right? And so it's just this enzyme called aromatase that modulates their activity, like that. Yeah?
Are the genes methylated to prevent or transcription?
So, I mentioned that we'll talk some about DNA methylation, like that. And that's something that can be dynamic, that can be changing. And for this example, are there changes in DNA methylation when these animals change sexes? I don't know. But and you'll see in like this class, in other classes, but especially this class, I just Google and then I just use an AI overview, and I could dive into that, right? I could look at the science papers. But yeah, you're welcome to just be doing that in class and being like, nope, or yep, and like that, and just be, like that. It doesn't bother me if you're doing, whatever you're doing on a device doesn't bother me. We just try to respect other students, and not restrict them. So that's this. And we'll see that, there are other types of sex changes in vertebrates. And our book talks a little bit in the first chapter and more in the second chapter about reptiles. So crocodiles, maybe I think alligators too, their sexual development is based on temperature. So at a different temperature. And they essentially have aromatase activity that's temperature dependent. There's concern, right, with climate change, of whether or not we'll be able to have reptiles around because it's getting warm and they're not making both sexes, they're making more preponderance of one of the sexes, like that. And so just, startling facts of life, right? That it's just, the conversion from testosterone to estrogen. And I always just say testosterone and estrogen. Estradiol is a specific molecule of estrogen. A lot of people say androgens instead of testosterone because testosterone is a specific molecule of androgens, like that. But just to show you a little bit of that, okay? And so then the next thing to show you in the second chapter, just to get, lead toward that, is that enzyme aromatase is present in different levels in different parts of the body. So, for example, in the brain, there's a small number of specific cells that are converting, and I can't remember right now, but converting, I guess, testosterone to estrogen. And so when in a male, you're like, this is all testosterone, a lot of testosterone stuff. It turns out that certain male behaviors are actually due to estrogen because it's produced or converted, we would say, from testosterone to estrogen. And that's actually what's modulating that, what you consider some really classic male behavior, like that. So, lots and lots of knowledge about some of the simplicity of sex differences and some of the simplicity and some of the complexity. Yep, and again, just start to introduce it. So the next thing we want to say is that the role of both of those are to activate gene expression. And so we'll see, we really want to look at changing gene expression because that's what those guys are doing, is they're changing gene expression like that. And maybe, increasing or decreasing aromatase activity or production, but also all kinds of other transcription changes, gene expression changes. Like those pigments, producing the pigments, changing from egg production to sperm production and stuff like that. And so we'll learn about the receptors for those hormones. And so estrogen and testosterone are both steroid hormones, and their receptors are an unusual group of proteins, a family of proteins that are transcription factors. And so these are both really famous, right? We really want to learn about testosterone and estrogen. And then when we say, well, what do they do? you know, so you're saying we're changing from testosterone to estrogen. What's that? How's it gonna occur? And the answer is we're gonna see changes in gene expression because the receptors for these guys are in the nucleus, and they're in a family of proteins that are called nuclear receptors. So we're just, you know, this is chapter 2 topics, but we're framing why did they pick that fish sex change? And the answer is because it just opens up. And in our bodies, the nuclear receptors are a gene family that's so varied, people call it a superfamily. But the nuclear receptors all have the same, similar, same mechanism of action. And in chapter 1, many of the topics we talk about are involve hormones, not just estrogen and testosterone, that act through nuclear receptors. So we'll talk about frog tadpoles and how the tadpoles go through metamorphosis to make a frog. And that hormone that does that is thyroid hormone. And the thyroid hormone receptor is a nuclear receptor. Thyroid hormone is not involved in our maturation, our metamorphosis, but it is in amphibians. It has a role there, and they study that thyroid hormone nuclear receptor. You'll see that a big part of stories of natural plasticity have to do with stress response. And you're familiar with acute stress response, like, you know, if a big old tiger came into the room, we would have a big rush of adrenaline, of epinephrine, right? And we'd be like, whoa, get out of here. There's another kind of stress response, and when we talk about the stress response in our course, it's a chronic stress response. Like if you catch a wild animal and put them in a cage, they have a chronic stress response, and that's due to an increase in corticosteroids. And glucocorticoid receptor is the receptor for them. It's another nuclear receptor family member like that. And you know, Luke asks, is there changing DNA methylation with the estrogen testosterone story? I don't know. There is with the glucocorticoid receptor story. There are changes in the methylation of it and other steps along its pathway that are based on that. For example, yeah?
I was just going to say, does it do that on purpose? Is it programmed into the nuclear receptor, or is it by accident?
You mean like the changes?
The methylation.
Yeah. So definitely not by accident. So here's an example of that, and we'll talk about the stress response because the stress response can lead to developmental differences. Here's an example that I don't know is not necessarily one that's really accessible in our course, but if you look at identical twins, well, they have the same DNA sequence, and so as they grow up, you know, same DNA sequence like that. And they have a DNA methylation pattern. And if identical twins experience different environments, the DNA methylation pattern changes. And so one study showed that, to answer your question, Mike, in humans, if you look at the glucocorticoid receptor gene, so the actual gene for the receptor, identical twins who are adults will have different DNA methylation patterns of that gene if one of them was systematically bullied as a child. So they have modulated their lifelong stress response because this is on that pathway of how to respond to stress. And my understanding is the interpretation of that is, and we want to be careful not to, say, oh, I learned this in class, because it's like the interpretation. But the interpretation is, I'll say it kind of like this. So, you can have, because my daughters get lots of pets, right? And one of my daughters gets rescue rabbits. And they are always wound up. Rabbits are always wound up, right? They're a prey animal. But she's had these rabbits for their whole life where they're just freaking out. You know, like if you come into the room, that rabbit is just flying around in a cage going, oh, I'm out of here, right? So what's going on with that? And, you know, I suspect it's a thing where that rabbit had a really tough, you know, childhood, we'll say, and has gotten modified that chronic corticosteroid dependent stress response permanently, like that. And we talked a little bit about that, is the idea of that is a predictive adaptive response, you know, where we'll see in a lot of development, the organism has some plasticity, and it will make a permanent plan, and just being like, I guess I'm living in a place that's really stressful, and I gotta be ready to just jump and run at any moment. So, you know, again, not fact, but that's an example, because you said, is that random? Let me say, you know, that somebody asked me, and I should share this with everyone, just to make sure nobody gets confused. So our course is divided into these two halves. The first half is the natural plasticity that occurs, that's evolved, it's part of the species and stuff. And then the second half is a disrupted one, like a pollutant or something, in the environment causes a disruption like that. And your project can just be in one or the other of the categories. The student asked me, do we have to cover both parts of this, right? And you do not. And so you could pick one that's all about the second half, the disruption and issues, problems, or all just natural plasticity in the first half. And you should, right? You shouldn't try to do both. That would be weird and just hard. However, some of the mechanisms that we talk about fit into both. So right now we haven't been talking about DNA methylation, and we'll talk more about that. So if you maybe haven't heard of that before, we'll get into that, because we are kind of doing with the chapter 1 and saying, why are we picking these examples? Because they lean into the mechanism like that, right? So let me just say this, that in the first half of our course, we'll talk a lot about natural plasticity. And for example, another example that is brought up in chapter 1 is social insects. And so like a queen bee and a worker bee are identical twins. And they're different because of the environment. And what happened is, when they were larvae, the queen bee was fed something called royal jelly by the workers. And there was a natural developmental plasticity that changed that identical twin to develop very differently, right, like that. So it turns out the royal jelly affects a hormone called juvenile hormone, and the juvenile hormone mechanism of action is to change DNA methylation patterns. So you can actually, you don't even have to feed a bee larva the royal jelly. You don't even have to have it do the royal jelly juvenile hormone thing. There are some nutrients that are essentially methyl donors, and you can actually just increase a methyl donor in the diet and get bee larvae to develop like a queen. Yep.
So in your two examples of the rabbits and then the bees, when that happens, like when they get the royal jelly, does it change the ligand, which then changes the nuclear receptor, which then changes the methylation, or does it just directly change the methylation?
I don't know.
Okay, fair enough.
Yeah, because there are lots of steps on the pathway, right?
Yeah.
And so the one, that identical twin one, is satisfying because you're like, oh, that's the gene for the receptor, so that's obvious. Then other ones would be like enzyme that helps synthesize a ligand, things like that, right? And so, you could have many steps that could be modified.
But ultimately, it changes the methylation.
Yeah, and so we're hearing that just, animals have evolved to have plasticity, and in some of the examples, that includes changing DNA methylation patterns, normal plasticity. So in the second half of our book, just to bring this up for the first time, the author wants to talk about DNA methylation for this reason, okay? So when our bodies, an animal body's growing, right, we grow and the cells divide, We gotta replicate the DNA, and there's really good DNA repair mechanisms, and really the DNA polymerase enzymes that copy it have really good accuracy, really good fidelity. We don't wanna make mistakes when replicating the DNA. It turns out that when cells divide, they also have to copy their DNA methylation pattern, and those mechanisms are not as accurate. And so if you look in like a person, as they age, you just start to see some kind of randomness in DNA methylation pattern. And that leads to defects, in cell function. And because our author wants to pitch it into that area of lifespan, and saying, part of aging is just this natural process of having, inaccurate DNA methylation, and you start getting, out of whack like that. So just to say that, we'll mention that a couple of times, but we're not looking for that. We're not looking for examples that we like, no, this fits through our whole course. But it does show that we'll talk about DNA methylation in our course a lot, okay? So these receptors, so it turns out, that this nuclear receptor superfamily sits at the center of a lot of modulation. And it's not surprising because they're hormone receptors, and we'll see hormone receptors are a big deal in this thing, because if you want to switch things up, having a hormone regulate the overall body plan is good. And so you don't have to scramble to write these down, but I wrote down some of the nuclear receptors, and these are nuclear receptors in vertebrates, and you'll see from chapter 1, the author talks about several examples of insects doing normal developmental plasticity, and when they look at what the mechanism is in the insect, it really comes up often that nuclear receptors of insect or arthropod specific hormones come up. I mentioned this juvenile hormone receptor, for example, like that, okay? So again, just kind of leading into that. And one of the concepts we want to understand is the concept of an ortholog. So human hemoglobin and dog hemoglobin are doing the same thing, and if we look at the gene sequence, it's really, really similar, and that's called an ortholog. So we understand at the organismal level that humans and dogs have a common ancestral species. Well, that's true if we drop down to the gene level as well. So because of that, we can also roam across huge, varieties of species in these stories. I said, well, all vertebrates have estrogen and testosterone. They all have estrogen receptor, testosterone receptor. They're all working the same way. And so we could find out something in that fish that would possibly have medical significance to humans, because we're all closely related like that. Now, insects don't have estrogen and testosterone, but they have those nuclear receptors. So when we say, oh, this nuclear receptor family, that covers a lot of territory and animals like that, okay? And so we can talk some more about that, like that. So just an example of that, and we'll get into that some more, okay? So that's that fish, and we just picked that for that reason. And so what we talked about is there's gonna be a change in the hormone, and you've heard about that, okay? So now just picking on the fish, I also just want to introduce another major piece of our course. And that is, think about this. So all of the females are constantly monitoring their environment, apparently through looking through their eyes and seeing if they're the biggest female and seeing if there's a male, right? So that's the apparent stimulus for sex change in this story, right? Want to think about that, right? I don't think, the male is not producing some chemical to, be showing that it's present. It's a visual thing, like that. And maybe behavioral too, right? So they're seeing this aggression and they're like, yeah, that's not me, or something like that, right? But also the size thing, right? That it's going to be the biggest female that's doing that. And so that brings up this for the first time this semester, which is another big deal, and that is that the brain can integrate a lot of environmental information. Really? So that's on the pathway, I would say, of this story, is the brain. And so you'll see, we want to talk a lot about the brain and how the brain is going to adjust hormones. Okay. And I'll just show you a couple of slides because we'll dive into this more when we talk about the second chapter. Our author doesn't talk too much about this, but just as, for example, is there's some cells in our body that are neurons that are called neurosecretory cells, and they release molecules that are either local hormones or systemic hormones, and they're really guiding the estrogen and testosterone synthesis in the gonad. So most of the estrogen and testosterone in vertebrates are synthesized in the testes or the ovaries, and it's these neurosecretory cells that are on the brain, like the pituitary and hypothalamus. And we can simplify that. It gets very complicated, very quickly. We can simplify that, and we want to think about that. And so you'll see in our course, there's kind of a buzzword that we'll refer to pretty often, and it's called it's spelled like this. It's spelled like neuroendocrine axis. So that's a really common term, and people usually pronounce that like neuroendocrine, and they're talking about that really tight relationship between the nervous system and the endocrine system. So what we've done here is we said, oh, these fish change shape, change sex, and so we immediately go to the hormone. And then we go to the stuff downstream, those nuclear receptors, and they're going to change gene expression like that, right? And we'll do that in the amphibians with the thyroid hormone. Oh, it's got a nuclear receptor, and let's start looking at genes and gene expression. And we'll do that with that corticosteroid stress response. Glucocorticoid receptor, nuclear receptor. And we go downstream of the hormone. But we also want to talk about mechanisms upstream, and very often we'll want to talk about this neuroendocrine axis. So because the brain can be interpreting the external environment through sensory organs, but it can also be interpreting internal status in different ways like that. And so that becomes a big deal for this plasticity is, how are you monitoring the environment? How are you integrating the environment status into the body through this neuroendocrine system axis? And so just to show you that I put some of the later slides in here just to show you. We got to talk some about the nervous system in our course and mechanisms of having those pathways work, and how do neurons control the conversion of estrogen, testosterone to estrogen and so forth like that. How does the brain do that? So some of that, and so just some slides about that, and we'll get back to those, right? So here's the term, the neuroendocrine axis. Fundamental integrated network connecting the nervous system, endocrine systems, primarily this, blah, 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 like that, right? So just a little of that awareness, right? Because that's fascinating, right, to think about, right? What is that fish doing? And it's constantly monitoring the environment, paying attention to what things are going on, and ready to adjust things, and it will do that through these neurons called neurosecretory cells. So we got to learn some more about that. So great. So that's just our first example, and our Monday discussion is going to be you bringing up examples of this plasticity that you found in the book, or that you think, is that happening in nature? Is that what that is, right? And it could be plants or animals. I'm an animal biologist. The author of most of the literature we'll end up reading is authors are animal and tend to be the animals. It turns out plants in this are even better because they can't run away. So there's tremendous phenotypic plasticity in plants like that. So that's what we're looking at for that, okay? So I just want to show you now, there's a slide in the first chapter about those reptiles, right? And their sex determination based on temperature, and that's an environmental factor like that. Then here's an example of a polyphenism in insect, and this is a butterfly that lives in Africa. And in its environment, there's a dry season and a rainy season. And this is the same species, and it looks different. The wings look very different. The left is the dry season, the right is the rainy season, like that. And it's the individual animals don't change. They're just reproducing throughout the years, throughout the seasons. And if a caterpillar is born in the rainy season, it grows up to be a butterfly that has these beautiful eye spots. If a caterpillar grows up during the dry season, it grows up to be a butterfly that has no eye spots. And so that's a part of how, insects mature. We just talked about, some about vertebrates. Insects are going to go through larval stage and then go through metamorphosis to an adult. And there's lots of this plasticity, and so there are examples of that. And it turns out that the scientists who studied that, and it's talked about in the second chapter, they've looked, and the thing that's different in the body is the level of a steroid hormone that's not in our bodies, but is in all arthropods. And that steroid hormone has a nuclear receptor. So, it's like, perhaps, it's got nothing to do with us, and it does not have really anything to do with us. But when we look at the molecular mechanism, it's like, oh my gosh, it's another nuclear receptor story. And we can start with the hormone and look downstream of it and ask questions about DNA methylation and gene expression patterns and stuff like that. And we can look upstream and talk about, how is the hormone modulated? It's made in an endocrine gland. What's regulating that endocrine gland? And some of the examples we'll have is, it's an environment that's being monitored by the nervous system. And it could be the external environment or the internal environment like that. Yep, so just to show you a little bit of that, why these picking. And see, here's a table. You don't have to scramble to write it down. But it's just a whole bunch of insects, and they all fit into the same thing. All different stories, like that, and they fit into, go inside and let's get started, like that. So here's another one, and this is a bedtime story, so we, have another 15 minutes. This is a great, I literally learned about this when my daughters were little kids and wanted to hear bedtime stories. And there are two girls, and you'll see it's just great. It's like, here's the dad teaching his little, kindergarten-age daughters what life is going to be like, okay? And I was talking to Luke about this last week because we were talking about topics. But there are these dung beetles, you may have heard of them. Dung is poop, and they will be out like in a field where there are livestock or big animals, and the male will roll a ball of dung around. And that's going to be used for food for the larval grub babies. And there are a variety of species of dung beetles, and they have different strategies. And these are reproductive strategies because the male is going to behave in a certain way to try to encourage a female to mate with them. So this type of species of dung beetles that we'll talk about are ones, and this is from the book, is one where the male does this. The male rolls a dung ball and rolls it away from the dung and then digs a hole and then puts the dung ball at the bottom of the hole and then guards the hole and does epic battle with other male dung beetles to keep them away from the hole. And a female comes by, that male mates with the female goes down and lays eggs in the dung ball. So that's the sexual reproductive strategy, is to make, essentially kind of like a nest of nutrients and then defend them. And they have these horns, and they use these horns to fight, and they use them to flip other males away. And, it's kind of famous that an ant can lift up, like 50 times its own mass. These, I think, are understood to produce the biggest force per mass ratio in the animal kingdom. They're just super, super rowdy, like that, doing that. So there you go. That's the reproductive behavior in this species, okay? All right, so now next thing is, just like every other animal, you can find some small males and some big males. The adult males are smaller or bigger. And that's just, could be based on genetics, right? I'm tall, right? My family, I grew up in tall people. Or it could be nutrients, environment, like that. So, we don't, mixture of those. But it could be nutrients, where a part of the size range of the species is just, here's a smaller adult dung male beetle, just didn't have as many nutrients, like that. So that's a kind of phenotypic plasticity. Then the next thing to say about that is that you would strongly predict that those dung beetles that have to be smaller would have smaller horns, like that. Then the next thing is that there's just a real common principle in animal biology, and that is you could make a graph, XY graph, where, I can just show this on this, just gonna make up a graph here. You can make a graph, and on the x-axis it could be adult male dung beetle mass, and these are the adults, you know, the adult males. And then on the y-axis it could be horn size, horn length, like that, okay? And the real strong prediction is this, it's called allometry. And that is, there's a scaling relationship where a smaller beetle will tend to have a smaller horn, like that. And that's just broadly true in life on Earth, right? I have long arms, my parents were tall and had long arms, like that. And it's not a perfect relationship because part of it's genetic, part of it's nutrients, and you know, it's just not a perfect relationship. So it's a scatter plot. And when you look at this particular dung beetle, what you see is this, where that scaling relationship is true for the biggest dung beetles, and then it's like sigmoidal, like that. So the smaller dung beetles are said to be hornless, and the larger ones are horned. And there's a threshold. So this is a kind of phenotypic plasticity that looks like a polyphenism, horned or hornless. And we've already talked about the behavior of the horned males. And the behavior of the hornless males is called, they're said to be, they have a different behavior, and they're said to be sneaker males. And there are lots and lots of species that do this. For the smaller males in that species, they'll have a different reproductive behavioral strategy. And so, that's shown here. I described what the horned males do, and it's a real significant behavior. They roll dung balls, dig a hole, put the dung in there, and then do epic battles with their horns to fight other males. And a sneaker male just cruises around and looks for a male that's guarding a hole and just digs a sneaker tunnel. Never fights, digs a sneaker tunnel over here and waits. And when the female comes down, the sneaker male mates with her, and he has a way to actually displace the horned male's sperm. Like that. And so what's the key principle here? The key principle here is the concept of, I see you want to make two different bodies, and you have two different behaviors, and they're instinctive behaviors. So you need to make two different brains to have the two different behavior programs, and you really want to make sure that the behavior matches the body. So that's what's going, why are we talking about this crazy example? And that is because you got to have a way to coordinate the animal program. This could be the same genotype, right? A single genotype could grow up to be a horned male and behave like a horned male, or grow up and be a sneaker male and behave like a sneaker male. Yeah? And the real difference is nutrients, that's the environmental difference, right? And a predictive adaptive response like that. And we better make sure you match them up. And the way they get matched up is with this steroid hormone. And the levels of the steroid hormone guide both. So just the amount and duration of the steroid hormone guide both. And it's this ecdysteroid, we'll learn about it in chapter 2. But see, it starts out sounding like literally a bedtime story. Because, right, my daughters to this day will call some guy, some dude, a sneaker male, like that, right? They'll be in a club, and they'll be like, he's a sneaker male, there's no doubt.
Oh, that's so true.
No, we don't want to go there. And, but, you know, yeah, and so they, you know, I programmed that into them. It's like, okay, listen up, this is what the world's like. But the key thing for us is you gotta have them coordinated. You gotta match up the behavior. The brain build has to match the hornless strategy. Because if it doesn't match up, that's not gonna be successful, right? A hornless male digging a hole and fighting, you know, like that. It's not gonna match up. So it's fascinating. It's a steroid hormone. They can create, you know, they can expose the larvae to different levels, different durations, do that. The hormone is one that we mentioned is found in arthropods and is a steroid hormone with a nuclear receptor. So downstream of that hormone, right, we got the gene expression, you know, and maybe DNA methylation changes, who knows, like that, but stuff we can learn about. Then upstream, who's controlling that? And it turns out the gland that makes the steroid hormone has a secretory cell connection. There's a cell in the brain that releases a hormone that stimulates the gland to make the hormone, and for how long? What's the input from the environment? In this case, it's nutrients. So in the fish, we have the brain integrating, you know, I think, visual and behavioral stuff. Here, the brain is integrating the nutrient status, which is more of an internal environmental thing, probably like that, right? So upstream and downstream from the hormone. We'll hear lots about hormones in our course, and we'll be like, whoa, what the mechanism upstream and downstream? Yeah, and see, and it's just a kicking natural thing that's happening. Like dragonflies have sneaker males. So big male dragonflies go out, they hover over like a pond, because that's where the females are gonna come and lay eggs. And the big males are, big adult dragonflies are really aggressive. And hiding in the weeds are these sneaker males, little tiny, same species, dragonflies, and they behave differently, they don't wanna fight. They also look more like a female than a male. And they'll just dart out and hope to mate with a female, you know, over the pond, like that, yep. Yeah, so, you know, it starts out super weird, where you're like, hey, you want to talk about dung beetles? You know, and you're like, no, I'm good. You know, not necessary. So that's an example. So it turns out that this example is really this, it's the same, it's definitely the same hormone, and it's an example they do the switch in a lab with levels and duration. They can do the switch between the dry season and the rainy season with that same hormone. Because one of the things about insects is they're going to go through this metamorphosis, right? They're going to really build a new body. And so they can integrate stuff that happened in the larva, the caterpillar's life, right, into that metamorphic plant and potentially produce different. So there's some really classic examples. That's why there's, where did I leave that? I lost track of it somewhere. Oh, it was maybe right after the butterfly wings. That's why our book uses a lot of insect examples and has this table. And look at here, mechanism, and it either says ecdysteroid, that's the hormone with the steroid that we've been talking about, or this juvenile hormone, the number one function of that is to modulate the ecdysteroid. And then there's an example of stress hormone, right? So see, you get into it and you're like, okay, this course is a lot about hormones. It's not all about hormones, but it's got a lot of hormone stuff in it, you know? And then, you know, not, you know, like down to the principles, right? And so, you know, just, you know, lots and lots of examples like that we'll talk about. Here's a graph just showing, you know, see that it's the distribution is not, it's very, you know, scattered. And the threshold is not perfect, but you can see it's not linear. Like that, right? Hornless and horn, like that. And just aside, we'll get into more, talk about allometric and isometric growth in our course, like that, okay? And some of these principles we'll get back to like that. But here it is, they're manipulating the steroid level in the animal just to change it like that. And so I mentioned the social insects, and DNA methylation is going to be a part of that, so we'll talk about that. And then, you know, we have a few more minutes, a couple more minutes, and the book talks in chapter 1 about amphibians. And so really different hormone. I mentioned it's thyroid hormone, so very different hormone. It's not in insects. It is in all vertebrates. Thyroid hormone plays a huge role in our life, especially fetal development. And it just happens that in amphibians, it's thyroid hormone that controls that metamorphosis. So when a tadpole grows up to be a frog or a salamander, that's thyroid hormone that's guiding that. So frogs, amphibians have testosterone and estrogen, and they're controlling sex determination. This thyroid hormone is controlling this metamorphic change. So the tadpole is going to really change its body, and that's thyroid hormone, and it also works through a nuclear receptor family member, the thyroid hormone receptor, like that. And so this is just showing that tadpoles respond to their environment. So the situation is amphibia have to go back to the water to reproduce, so mom is going to lay eggs in the water, right? Mom is hopping around and running around on land and is going to mate and go lay eggs in the water. And the mom can't judge that aquatic environment very well, so famously you'll see eggs in the spring around here in puddles. You know, she didn't even find a nice big old pond. They're called vernal pools. Yeah, they're going to dry up, and there could be signals in there of trouble. There could be a predator in that little pool like that, or the pool could literally be just drying up and shrinking, or it could be crowded. And so our book talks about that because there's the environment, and the frog is going to integrate that. So different ways of detecting it, maybe with their brain, maybe other features of them. And then it's going to control that thyroid hormone, which works through a nuclear receptor, and you'll have downstream changes in gene expression, like that. And so, for example, and there are different frog species, salamander species have different types of this that they've evolved. And so one quick example, and then we'll quit, is you can grow a certain frog species in like just an aquarium, just a fish tank in a lab. You can have just a mesh net so that tadpoles can't swim side to side, but it has the constant volume of water in it. And you can just move the net over, that kind of mesh wall over, so that the tadpoles have a smaller area they can swim in. And if they get down below a certain threshold, they'll accelerate their metamorphosis and turn into a frog quick. Because they'll literally say, I'm not able to swim very far before I have to stop. And that's making me think I better get out of here. It's not drying out. There's plenty, it's not like it's, the volume is not changing. It's just their ability to perceive the volume is changing. Isn't that wild? Like that. But it goes to the idea of, well, maybe that, it's a bad environment like that. So we'll talk more about that. But you can see, the author is not just haphazardly grabbing examples in chapter 1. And what we want to do for Monday is really just kind of have kind of fun with this first chapter in saying, whoa, this is freaking me out. Like talking about sneaker males and other things. Find some examples of the book you want to talk about, or questions, why are we talking about this in this course? Or dive into some of the mechanisms, or find something online. Or, like, I'll bring my caterpillars from lab, because it happens that my research fits into this course. I could bring my caterpillars from lab and